WPPC, as you all know, whey protein phospholipid concentrate, is a dairy byproduct of the dairy industry, and it contains a large portion of WPPC is phospholipids, and phospholipids, when ingested by human, is positively associated with better cognitive function. So today I'm going to introduce from waste to wellness upcycling dairy byproducts, WPPC, for a healthier brain. So good morning, everyone. My name is Kevin from the Bolling Lab at UW-Madison. So, guys probably see this too much today. So allow me to briefly go through it. So WPPC is a an end product, a retentate of the cheese processing after the ultrafiltration and microfiltration. And WPPC, its main ingredient is milk fat globule membrane. So you can see to the right, it's a WPPC powder here. And it's also here. So if you guys are interested in that, welcome to see my poster, and you can try it, you can have it too. It's edible, just in case. So let's go back to this figure. WPPC powder, take a closer look. It's actually, there's a special triple layer of the phospholipids of the milk fat globule membrane, which I will use the WPPC instead for the following. Triple layer of the phospholipid incorporating the triglyceride with the protein and glycoprotein and others attached on it. So we say that WPPC contains a lot of phospholipids. So what's actually in the phospholipids? So they mainly contains the phosphatidylcholine and phosphatidylethanolamine and other phosphatidic acid and choline precursors here. So where can we find the choline, phosphatidylcholine? You can find it in the food everywhere here. And choline is a vital nutrient of human and involves in several different metabolic pathways involving like lipid metabolism, gene expression, cellular signaling, that kind of so on and so forth. But for this presentation, I want to focus on the cognitive function and brain development. We can find the top three of the choline existing in the food is organ meat, egg, and milk. And how can we find choline? What's the main sources of human body to obtain choline? So dietary sources, that's what in the previous slide. So when we ingest dietary choline, those choline comes into like phosphatidylcholine and other phospholipid precursors, like what I was mentioning. And sometimes it can break down into these three kinds of the metabolites. And it can transfer into liver and go through this cycle we call the one-carbon cycle about the gene regulation. But the other side, we actually can synthesize the phosphatidylcholine by ourselves. So now we have dietary choline as a main source and also the hepatic biosynthesis. But unfortunately, from 2017 to 2020 NHANES, which is a National Health and Nutrition Examination Survey, only 12% of US population, including men and women, meet the adequate intake of choline. And look at the figure here. So the dots here are the adequate intake of each age group. And the bottom, the bar is the regular, like people, how much people eat in their regular diets in the survey. And we want to focus on specifically on the age between 51 to 70. I'll explain that why. Only 8% of women in this age group consume enough choline. That means adequate intake. And when the committee in the dietary guidelines for American, they established this number. When they establish adequate intake, that means they don't have enough data to support the numbers, or they will form like a recommended daily allowance, which is RDA. So you can imagine for all this part, maybe it's not correct, or maybe we don't know for the current scientific community. So we think that post-menopausal women probably need more choline. But I'll explain that why. So please look at the left figure. So estradiol E2 is a major component of the estrogen. And the y-axis is the amount of E2 production. And the x-axis is the age. So you can see the purple means the amount of estradiol. It declines with the age, with aging. So that means postmenopausal women has less estrogen level. And please look at the right figure. So remember, this part is about the biosynthesis of the phosphatidylcholine I mentioned earlier. So we need CDP-choline and diacylglyceride. Also, CDP-choline actually comes from the breakdown of the cell membrane. So we need these two ingredients to make the phosphatidylcholine with the help of the enzyme called PMT. The PMT actually heavily regulated by estrogen level. So when the estrogen goes down, it's going to negatively regulate PMT, and as a result, phosphatidylcholine's production is going lower. So what's the significance of that? We know that the phosphatidylcholine status is positively associated with the risk of the cognitive impairment in a previous study. And 32% of Americans aged over 65 years old actually have some degree of cognitive impairment, and it will cost us $355 billion in the healthcare system annually. And that only accounts for the Alzheimer's disease. We didn't talk about the dementia, mild cognitive impairment, and other cognitive function-related illness. And let's look at the WPPC. WPPC has been considered as a waste of the dairy industry. And when the industry makes the WPPC, they cannot just take the WPPC out and dump it on the landfill. They have to process some treatment on it. And it costs 1.9 cents per gallon to treat the WPPC. And then imagine we have 30 to 35 million tons per year every year, worldwide. So wouldn't that be great if we can get WPPC to use to benefit people's mental health, cognitive functions? But the major ingredient bioactive of the WPPC, phosphatidylcholine, is not always good. In the previous study, we see that free choline and actually metabolized into TMA and TMAO in our gut. And TMAO has been considered as a detrimental factor that will positively associate with the increased risk of the cardiovascular disease and actually other diseases too. And TMAO and also activates the NF-kappa pathway, which associate with the pro-inflammatory pathways. So what's our project goal? Is that we formulate the WPPC and ask people to drink it. And we want to see if the WPPC really deliver the choline into our bloodstream. And also want to see if the WPPC will generate the TMAO, which is a detrimental Then, since this pathway actually involves the gut microbes, so we want to see what's the impact of WPPC to the gut microbes. And does the gut microbes actually can process the WPPC and generate the beneficial compounds called lysophosphatidylcholine. And it has been associated with better cognitive function. So our study design is that we are going to enroll 24 postmenopausal women divided by, randomize them into two different groups. One is WPPC. The other one is whole egg powders group. And the reason why we use a whole egg because whole egg consists of a single layer of the phospholipid. But WPPC has triple layer, 3 layers. And whole egg has been considered as the most bioavailable choline for the current standard. And we want to see if our WPPC is better or just similar to the egg. So For the day one, they will go through the informed consent process and we'll do the biometric survey. And they consider as enrollment if they agree to attend. And after the 30 days, for the following 30 days, we'll ask them not to increase their weight by intentionally increase or decrease their weight, and not to consume the supplements regarding choline or its cofactors inside. And after that, we'll ask them to go through diet record. We use ASA24, which is a 24-hour recall, to let us to have a better idea about what they're actually eating. And after that, we'll ask them to collect their fecal samples and the urine, 24 hours urine samples, and they can come to our study site for a visit. And by then, we'll collect their blood too as a baseline. And we also want to collect their blood pressure, waist circumferences, like height and weight, like basic anthropometrics. And then we'll ask them to bring back 10 packets of either WPPC or whole egg pouch, and ask them to formulate it with 2% milk and drink it every day for the following 10 days. And they will come to our study site to do the same thing as the second visit. So how do we detect the differences? How do we decide the bioavailability and if there's safety to drink for the WPPC? So our primary goal is to see the choline and its metabolites actually exist or increase in the blood circulation. That means we successfully metabolize it and absorb it. And so we'll use the LCMS to achieve that goal. And also, we want to see if the WPPC is safe to consume. So we use ELISA to detect high-sensitive C-reactive protein and other pro-inflammatory cytokines. And then we want to see does WPPC have any impacts on the gut microbes. So we are going to do 16S RNA sequencing to see the general population change in the phylum level. So our expected outcome is that the increase of the circulating choline in both WPPC and egg groups, but probably WPPC is better. And we want to see if the TMAO is not generated and other pro-inflammatory cytokines is not generated in both groups. So since this study is not designed for doing the cognitive behavioral changes, so I'll leave that for the next study. For now, this study is detecting the bioavailability and the safety of the WPPC. So I want to thank my teammates in our Bolling Lab and our collaborators in Austric Lab. And I want to thank the DIH, NDC, and the Lakes Foundation for the funding support. That's my reference. And if you are interested in that, you can scan the QR code here to our lab. And if you want to take a deeper look on what's going on, we register our clinical trial in clinicaltrial.gov. So please scan that too. Or if you just want to chat, welcome to send me an email. I would love to discuss with you. And thank you for your attention.
Questions for Kevin?
For the study you're planning, what kind of end value are you looking to get to? Like, what is your goal of participants that you're looking to achieve? Like, do you have a goal in mind? I'm just curious.
Oh, you mean numbers of
participants? I'm like curious, like the scope.
Oh, it's a relatively smaller study. We only have 24 participants, but it's actually powered. So that's a nice thing.
We have time for one more question. If anybody has one.
Do you think that the choline is going to be easier for people to digest than milk or egg, or I mean, is that the goal to find out?
Yes, we did have a sensor test about that. So hopefully, I know we are still working on the data processing. But if you want to try, it's here. Chocolate flavored.
Okay, that concludes our session for today. Thank you, Kevin, very much. And thank you to all of our speakers. Thank you.